Dr. Wei Xie. He is a professor at Tsinghua University. As I mentioned earlier, he graduated from Peking University as a bachelor degree of science undergrad student in 2003. Then he went to US, pursue his PhD study at UCLA, where he studied the function of histones and histone modifications. He also obtained a Master of Science degree in UCLA in statistics. Then he continued as a postdoc in epigenetics and transcription regulation at UCLSD San Diego in 2009. And then he joined the Tsinghua University as a principal investigator in 2013. Currently, he's using interdisciplinary approaches to try to understand how life begins after fertilization through chromatin and gene expression reprogramming. His group established a series of ultra-sensitive technologies to analyze chromatin dynamics and gene expression using only a few hundred cells. His work has illustrated how the epigenome is established in gametes and how it is transmitted and reprogrammed after fertilization, and how the embryonic epigenome and the transcriptional program are re-established.
Okay, cool. I'd like to first thank the organizer for giving me this great opportunity to share with you our recent research progress. And I have to say that this is a wonderful seminar across the globe for life science during the pandemic. While we cannot travel, but we can still enjoy science with the colleagues in the world. So thanks a lot. So today I'd like to share with you a recent story in our lab, which is related to decoding chromatin reprogramming and zygotic genome activation when life just begins. So I'd like to start with my favorite picture, illustrating one of the most fascinating phenomena in biology, I think, which is fertilization. So this is our long-term interest in lab. That is how the life clock is resetted every single time after fertilization. This sounds a crazy idea elsewhere, but for fertilization, this is a natural phenomenon that happens every time that we can see it in the world, every day. But of course, the question is how, right? And thanks to a joint efforts from many pioneers in the field, now we can understand that to reset the whole life clock, it takes a lot of efforts. That includes reprogramming of the chromatin or the epigenome. That includes, for example, DNA modifications, histone modifications, chromatin structure, and so on. But besides that, the embryo also have to restart the entire transcription programs. As shown in this figure, this is true for every single species that has been studied. Mouse, C. elegans, Xenopus, zebrafish, Drosophila. After fertilization, the whole genome actually stays silenced for a few cell cycles before it gets activated. It depends on different species. For example, mouse takes one cell cycle, but zebrafish doesn't start the zygotic genome activation or ZGA until about 1,000 cells. So this is quite interesting phenomenon. Taking mouse as example, and the zygotic genome activation occurs around the late 2 cell stage. This is in fact so-called major ZGA, and also the first ever transcription event in life. But before that, there's also minor ZGA, in which only less than 100 genes are actively transcribed and generating functional proteins. So this is quite interesting to us because why there's this special group of genes that are transcribed ahead of everybody else. So perhaps not too surprising, when the major ZGA is blocked, the embryos cannot develop beyond the 2-cell stage. Because the whole transcription program basically is shut down. There's no way for embryo to further develop. But the question is, what about the minor ZGA? Why there's this small wave of transcription before the major ZGA? And if this minor ZGA important or not? And in fact, work from Oki and Richard Schultz's lab have shown about 2 years ago, when they blocked minor ZGA using DRB, which is a reversible transcription inhibitor, temporarily, and then wash away the drug before major ZGA. Interestingly, these embryos still cannot develop beyond 2-cell stage, suggesting that minor ZGA is just as important as major ZGA. So then the question is, what's really transcribing in minor ZGA, right? It has been studied quite extensively recently, and a few genes turned out to be quite important. For example, ZSCAN4 is a DNA binding factor that functions in telomere elongation and also genome stability. As you may know, after fertilization, the telomere has to be elongated in order to reset the life clock. And the DUX4 is a transcription factor that can activate ZSCAN4, as well as a number of embryo-specific retrotransposon elements. In fact, this small group of genes are also called 2-cell-like genes, and they are also transiently expressed in a special subpopulation of multi-embryonic stem cells. And those cells are called 2-cell-like cells. So, but this is not the whole story. And in fact, Oki and Richard Schultz's lab have previously also found elsewhere in the genome, beyond this small group of genes, there exists a pervasive but very low-level transcription genome-wide. They are not polyadenylated, they are not properly spliced, meaning they are probably not generating functional proteins. So these fascinating findings have led us to ask a couple of questions. For example, is this promiscuous transcription during minor ZGA just a byproduct, or it actually may have functions? And also, similar to many people, we always wonder why major ZGA does not start right away after fertilization, but the embryos have to wait a few cell cycles. So what's really happening in this very unique developmental period? To address these questions, we have to understand how transcription is regulated in early embryos. But this is in fact not an easy task, simply because the limited materials that we can collect from research animals like mouse. So as a matter of fact, when I started the lab maybe 8 years ago in Tsinghua, there are not a lot of technology that we can actually use to study transcription in embryos, except we can look at the embryos using the fluorescence, but that doesn't really tell us what happens at the DNA levels. Therefore, we have spent a great effort trying to come up with different technologies to allow us to look at the chromatin regulation, not using millions of cells, but instead of 20 to 500 cells. And fortunately, we have extremely talented grad students, a postdoc in lab, and they came up with various methods as shown here, so that we can actually do the research using the mouse embryos to look at chromatin regulation and DNA levels. And so if you're interested in any of these technologies, please you're welcome to send us an email, and we can share with you our the most updated lab products. But basically, now we can study DNA methylation, his modifications, chromatin accessibility, 3D chromatin architecture, and even transcription machinery. So with these tools, we can now we decided to focus on the most central transcription machinery, which is RNA polymerase II, that can work with transcription factor and chromatin regulator to initiate the transcription of most genes in the genome. So when we look at the chromatin binding of PO2 simply by immunostaining, so here's what we can see. Polymerase II is strongly enriched in fully grown oocytes, which is still active transcribing. But later, it was kicked off chromatin in metaphase II oocytes and a one-cell embryo at the early stage, when transcription is entirely shut down. By the middle of the one-cell stage, PO2 comes back to chromatin, and by the late two-cell stage, the major ZJ starts. So you can see that the chromatin has a very dynamic binding of, the polymerase II has a very dynamic chromatin binding during early development after fertilization. This already immediately raised several questions. For example, when PO2 comes back to chromatins, since major ZG has not started, what's it doing on chromatin, right? Does it really bind promoters? Is it actually poised there without transcribing genes? And what is waiting for? Is it waiting for the maturation of chromatin? So these are the questions that we wish to address. But one simple way to answer this question is to do this so-called ChIP-seq experiment of chromatin IP to look at where PO2 bind to which genes. But again, as simple as it sounds, this is not an easy task at that time when we started the project perhaps 3 or 4 years ago. Again, due to the limited material that we can use. And this is when Bofen Liu, a very talented postdoc in lab, decided to take on this challenge, and by working with Qianhua Xu and Qiujing Wang. So together, they came up with their own technology, which is called STAC-seq, short for small-scale TIM5 assisted chromatin cleavage sequencing. This is simply, to put it simply, this is a very highly sensitive ChIP-seq method, so that we can look at the chromatin binding of proteins using just 500 cells. In fact, this method employs the similar principles as cut and tag, as nicely developed by Steve Hennekopp and also other labs using under different names. But at that time, we didn't really have those technology available, so this is our own version. And fortunately, it works very well for polymerase II binding detection. But basically, we generated the TM5 protein AG fusion protein in collaboration with Wei Xie, incubated with the antibody, apply the complex to cells so that the antibody can recognize the epitope while the TM5 will cut the chromatin. And then we can purify the DNA, which is already segmented by adapters for sequencing. So therefore, we can directly sequence these fragments so that we can know where polymerase II binds. So without going into the details, here's the results that we generate in mouse oocytes as well as early embryos. As you can see that polymerase II binds strongly to chromatin in fully growing oocytes, which is still active transcribing, but we can no longer detect its binding in metaphase II one-cell state, or middle one-cell stage. But by the end of the one cell, PO2 comes back to chromatin and stay there, as you can see here. So with this extremely valuable data sets, we can now start to ask questions like, where does PO2 initially bind after fertilization? Of course, the first set of genes that we looked is the major ZG genes, as shown here by the green box. As controls, we also included oocyte-specific genes, as shown here by the red boxes, as well as developmental genes, most of which are not really transcribed in early embryos, so they are largely silenced. But surprisingly, when we look at the PO2 binding and these 3 classes genes, they all show very strong PO2 binding and the promoter regions, as you can see here. However, at the late 2 cell stage, we start to see PO2 binding much higher for major ZGA genes compared to the other two classes. But what's more interesting is, well, we found that at this early 2 cell stage, we already see the increase of PO2 binding and the major ZG genes again compared to the other two classes. So indicating some kind of pre-configuration PO2 to major ZG sites, even before the transcription actually starts. So this is perhaps more clear from this heat maps analysis, where you can see that PO2 peaks, many of them are shared between 1 cell embryo and the late 2 cell embryos. However, a large fraction of PO2 binding peaks are specific to 1 cell, and before they are lost, while cells develop, the embryos continue to develop to late 2 cell, and many of these regions acquire PO2 when the cells reach the 2 cell stage. And obviously, at the early 2 cell stage, this is an intermediate stage, as you can see. So this is quite surprising to us, because it almost indicates that initially when PO2 lands on the chromatin, many of the targets are not right. And it just somehow it later find all the right targets to initiate the transcription at the late 2 cell stage. Then, of course, the question is how, right? We believe that the transcription factors probably play critical roles to guide polymerase II to find the right genes. So currently, we're actively looking into this issue, and hopefully we can identify the transcription factors that does that job. But on the other hand, we also realize that the pre-configuration period is also the time when minor ZGA happens. So another natural question is how or whether the minor ZGA might play a role in this process. To address this question, we just did one simple experiment by treating embryos with transcription inhibitor, DRB, and then followed the PO2 binding along the way. And in fact, this is quite striking. When we add transcription inhibitor, you can see that this pre-configuration of PO2 is already effectively blocked at the early 2 cell stage. And even by the late 2 cell stage, we can still see a lot of PO2 are retained at the 1 cell specific targets. While we can see some PO2 coming to this late 2 cell specific targets, but it's much weaker compared to the control. So this data indicates that the minor ZGA somehow is essential for this pre-configuration polymerase II before transcription actually starts. So then we can try to ask another question, that is this pre-configuration PO2 important or not? So to do that, we took advantage of the same experiment that Oki and the Hughes lab did, which is treat the embryos with DRB to block minor ZGA and then wash them away before major ZGA to let embryo recover, and so then the major ZGA can start. We call this minor ZGA transcription blocked embryos, short for MTBE. And then we followed these embryos with transcription analysis and also polymerase II analysis. And here's what we found. From our signal analysis, you can see that the major ZGA genes are activated still in these embryos, but not sufficiently. What's perhaps more interesting is there's a group of genes that are ectopically expressed, as you can see here, in these MTBE embryos. So after careful analysis, we realized that these genes are enriched for 1 cell PO2 targets, as well as for developmental genes, as indicated by polycomb targets here. So without showing the data, we also found in the 1 cell embryos, PO2 likes to bind promoters with high CG content, which are actually enriched for development of genes like transcription factor genes. So let me give you two examples. This is SMAT6 and BMP4 genes. They are normally not expressed by the end of the late 2 cell stage. However, you can see their ectopic expression in MTB embryos, and this nicely correlates with their 1 cell PO2 binding and the residual PO2 binding in MTBEs. And in fact, some of these transcripts can be turned into proteins. So what I'm showing here is Win3A is a critical developmental factor or signaling factors that's normally not expressed in late 2 cell stage, but in MTBE, the Win3A is expressed as detected by immunofluorescence here. While 5A2 is a major ZGA gene, it is expressed normally in late 2 cell, but expression is much reduced in MTB embryos. So as quantified by the result here. So these data indicate that the minor ZGA is important for the preconfiguration PO2, and it is also important for zygotic genome activation fidelity. But of course, the million-dollar question is, how does this happen, right? And we don't really have an answer right now, but I'd like to propose a model here. So right after the fertilization, PO2 will land on chromatin that includes part of the targets are correct, but the other half or even more than half targets are not right. So these are the 1 cell targets here. And the PO2 have to be relocated to the right genes, presumably guided by certain transcription factors. However, because the chromatin initially and these regions are inaccessible, so the transcription activity or the elongation activity of polymerase II, perhaps it's important to destabilize nucleosomes, open up chromatin there to further allow the recruitment of more transcription factor and chromatin regulator and PO2 here. So this means even when the transcription activity is still very low before major ZGA, it is important at least to stabilize PO2 here once recruited by transcription factor. And this will essentially form a positive feedback loops. Now, if we add the transcription inhibitor there, this will block these feedback loops. So PO2, even recruited to this region, cannot stay there. So we'll probably just hit and run. So we believe that the polymerase II and transcription are not just the machine that transcribe genes. They could be even potent chromatin remodelers to remodel the chromatin landscapes. So with that, I'd like to stop here and briefly summarize. We believe that the promiscuous transcription during minor ZGA is not just a simple byproduct of PO2 binding there, but it actually has important functions by promoting PO2 and chromatin preconfiguration. And finally, why major ZGA does not start right away after fertilization for every single species. And we believe this is a critical period to allow embryos to synchronize ZGA firing with chromatin and PO2 maturation. All right, so with that, I'd like to stop here and thank people in my lab, particularly Bofen Liu, Qianhua Xie, and Qiujing Wang, who are the leading force of this project. Of course, this cannot be done without our wonderful collaborators. Na Jie from Tsinghua University is our long-term collaborators downstairs. And Yu Yang from Institute of Biophysics provides very important discussions and for the technology development and the initiation of the project. And the Team 5 enzyme was made in collaboration with Weixian. And these are our funding agencies. Thank you very much. I'll stop here and take any questions if there are. Thank you.
Thank you, Wei. Fantastic talk on the initiation of life after fertilization And let's get right into the questions. Our first audience question is from Eric. Eric Chow, please go ahead and ask your question.
Okay, yeah. So I'm just curious about how the minor ZGA is maintained at a relatively lower level at the early two-cell stage. And yeah, is that regulated by the chromatin accessibility or like the causing of PO2 during transcription?
Right, so that's a good question. So let me make sure that I understand it correctly. So you're asking why minor ZGA transcription level is very low.
Yeah, how is that maintained at the early two-cell stage?
Okay, so here's what I think. The transcription activity of PO2 is generally very low before major ZGA. That includes one cell and the early two cell. In fact, this was it was showing that the elongation key factors of PO2, which includes the cyclin T1, I think, and the CDK9, are not present, or most of them are not present in the nucleus in one cell embryos when people do a staining. So this wasn't done by us, but by previous work. So generally, I think the polymerase II cannot elongate very efficiently. That's why most of the transcription is happening at the very weak levels. However, you have a great point that because as I mentioned, there are a couple of genes that are highly transcribed. In fact, in one cell and early two cell embryos, like ZSCAN4, DUX genes, it's unclear where the specificity comes from. Why those genes can be transcribed without any problems. There could be certain mechanism to recruit elongation factors specific to those regions. And right now, we don't really know what are the mechanisms. That this answer your...
Yeah, thank you.
Okay, thanks.
Thank you, Eric. Thank you, Wei. Okay, I have a technical question for you. So you describe a very neat technique to assess the distribution of chromatin binding proteins at a single cell resolution with the STAC-seq. But then there are also similar techniques out there, CoBatch, cut and tag, and in fact, they also use the TN5 transposase. So how do these techniques compare with STAC-seq, and is there any distinct advantage to using one versus the other?
Yeah, that's a great question. That's a question that we were talking about every day in lab. They are very similar, first of all. However, I think for reasons some of them we still don't understand, their effects could differ for different antibodies. For example, polymerase II, we tried to cut and tag before, and this for the version that we used, we couldn't get it working. And only our STAC-seq works. But for other antibodies, it also varies. So sometimes, perhaps that there's also a cut and run technology, also developed by Steve Penikoff and others. Sometimes one antibody works for cut and run, but not cut and tag, and sometimes vice versa. So my feeling is different methods could differ slightly in their details, and those details could matter when they are applied for different antibodies. So in our lab right now, we just try for every antibody comes in, we just try different assays and see which one could give the best results. So of course, it will be great if there's a universal method that works for every antibody, but at this moment, I don't think it is the case. But specifically going back to your original question, our method, which is the STAC-seq, takes less washing compared to cut and tag. We also do the pre-incubation of TM5 and antibodies before applying the complex to cells. So that's at least two differences that I can see from compared to kind of tag, for example. But again, I don't really know whether these differences make this the final effects. You know, whether they are the factors that makes the difference. Yeah.
All right. Thank you. That was a great response. Thank you. So we have another audience question. You please go ahead, unmute yourself and ask your question. Are you here? All right, I'm going to read out the question. In the minor ZGA, does POL3 also produce a divergent transcription? And the other question is whether the minor ZGA protein product impacts the following major ZGA.
Okay, yeah, I can see the question too. These are great questions too. We don't really know if POL2 is producing divergent transcription transcripts. I imagine that this would require strand-specific RNA-seq analysis, RNA-seq, which at least for most of our data sets, we don't have this information. And also the minor ZG transcription is very weak. I'm not sure whether the signal is good enough to see the orientations with that, for example, strand-specific RNA, but it's definitely worth trying. So yeah, so that's my answer. The second question, sorry, what was the second question?
Oh, I'm sorry. I think I might have. Yeah.
Right.
All right. The second question is whether the minor ZGA protein product impacts the following major ZGA.
Yeah, that's definitely an important question. So what we have shown is those promiscuous transcription seems to play a role, but they don't generate proteins. So only those handful of genes that can generate protein, like ZSCAN4, DUX. So far, DUX role has been tested both in cell lines and in embryos. The results are a little bit different. In cell lines, DUX, the transcription factor, has a quite large impact on, you know, so for example, the embryo specifically repeats expression. But in cell lines, there's no cell line that can mimic the major ZGA at this moment. So we don't really know whether it has a function in major ZGA. Now in embryos, this was shown by Yi Zhang's lab, also Sharon Gao's lab. Knockout ducks have a moderate impact on major ZGA and also minor ZGA. But the embryos, many of them are fine. I think more than half of the animals can be born. So definitely it's not absolutely required at this moment. Now the other genes are less well understood. ZSCAN4 is probably the most well understood, but it's mainly involved in telomere elongation and the genome stability. I don't remember data showing that it's important for major ZGA at this moment. So I guess the answer is right now, we don't really see at least a dominant factor from minor ZGA that could regulate major ZGA. I hope this answered the question.
I think you did. Thank you, Wei. Thanks for the response, and thanks for the very insightful talk on chromatin remodeling and its role post-fertilization. Thank you. And with that question, the live session comes to a close. However, as always, the discussion continues at the Slack forum. Please check the chat box. You've been invited to a Slack channel. Go ahead, join the channel. Our speakers will be there to join you, answer your questions, or to continue the discussion. From Life Sciences Across the Globe team, thanks to our participants, Hongwei, Ying, and Wei Xie. Thanks also to the audience for participating in the lively discussion session. Next week, please join us at the Marine Biological Laboratory at Woods Hole. And we'll hear from Dr. Caroline Albertin and Dr. Roger Hanlon. The title of their talk is Rapid Adaptive Camouflage in Octopus and Squid, from Genes to Behavior. We'll also hear about education at MBL from Nipam Patel. So do join us. And until then, it's bye from all of us here at Janelia Research Campus. Take care.